Beginning to come in, so we might just in the interest of time, we'll get started. So I hope everybody can hear me okay and can see everything okay. So good morning, everyone, to all our attendees. Thank you so much for joining us. My name is Vanessa Moore. I'm the Scientific Project Manager for Dementia Research Network Ireland, DRNI. We're thrilled to see you all joining us this morning. And of course, it's Brain Awareness Week this week. It's extra apt that our webinar today focuses on neuropharmacology of dementia and other brain diseases. I want to welcome our two speakers today, Dr. Andrea Kwakowsky and Professor Keith Murphy. Thank you so much for joining us. Before I hand over to them, I just want to say that we're hoping to have time at the end of the presentations for questions. So please put any questions that you have in the chat box or in the Q&A box. You can see them there at the bottom of your screen. Just put the questions in kind of as and when they come up. You don't have to wait until the end to ask it all. So just feel free to type in all your questions at any stage. I'm just going to double check as well that the chat box is activated. So I will now introduce our first speaker, Dr. Andrea Kwakowsky. You're very welcome today, Andrea, and thank you so much for agreeing to take part in the webinar. Dr. Andrea Kwakowsky is a lecturer in pharmacology at the School of Medicine at the University of Galway. Her primary research interest is in the molecular and cellular basis of brain function in Alzheimer's disease, Huntington's disease, and multiple sclerosis. Dr. Kwakowsky's lab conducts multidisciplinary research that aims to transform the understanding of the pathological mechanisms of neurodegenerative diseases and to find novel drug targets and therapeutic strategies and to understand the underlying pathology of diseases such as Alzheimer's disease. And to determine whether manipulation of specific neuronal pathways could be part of an effective treatment. In this talk, Dr. Kwakowsky will present evidence of the remodeling of excitatory and inhibitory neuronal networks in the Alzheimer's disease brain and other neurological conditions. And some of the promising therapeutic approaches will be discussed that are currently under investigation in her lab. So I'll hand over to you now, Andrea. Thank you.
Thank you very much, Vanessa. Just a moment to find the presentation. Yes. Does it look good, Vanessa?
Yeah, looks perfect.
Okay, thank you very much. So good morning, everyone, again. And thank you for the kind introduction, Vanessa, and the opportunity to present my work today. Recent DRNI webinars focused on amyloid and tau targeting therapies in Alzheimer's disease. So today I'm going to talk about other therapeutic options, particularly those that target neuronal network problems that are characteristic features of Alzheimer's, but also other dementias and other neurological conditions like autism spectrum disorder, epilepsy, and schizophrenia. The research in my lab focuses on neurodegenerative disorders, as Vanessa mentioned, and we aim to understand the disease pathology by using multidisciplinary approach and find new therapeutic strategies. We study many aspects of the conditions, including neuronal network disturbances, neuroinflammation, and sex differences. And all this research, importantly, is performed in close collaboration with patient organizations and people affected by dementia. What is dementia? Dementia is an umbrella term for symptoms of a group of diseases of the brain that always impair thinking abilities, often memory, and in many cases involve behavioral and psychological problems as well. Alzheimer's is the most common type of dementia and accounts about 60–70% of the cases. Alzheimer's is the focus of this talk, but therapeutic approaches discussed are relevant to other dementias and neurological disorders mentioned earlier. It is important to emphasize that Alzheimer's and dementia are not part of normal aging, and the changes differ in magnitude and scale. And memory problems may be masked in a daily life by compensatory techniques by the person and compensatory behaviors of family members, but also well-learned routines and environments. Over 70 million people worldwide are living with dementia and more than 65,000 people in Ireland. And this number, it will almost double every 20 years. We are experiencing a dementia pandemic, and finding therapeutics that can prevent Alzheimer's and slow or stop the disease progression is urgently needed. Importantly, none of the neurodegenerative diseases have a cure, and most don't have efficient disease-modifying therapies available to treat them, even after decades of extensive research. There are some common features of all neurodegenerative diseases, such as neuronal death and neuroinflammation and vascular dysfunction. And the research in my lab has projects focusing on all these aspects of the diseases. And also there is the neuronal network dysfunction that can precede, but also can be the consequence of these pathological changes. Alzheimer's is a neurodegenerative disease, as I mentioned, affects millions of people worldwide, but after several decades of research, there is no cure, effective, safe treatment to slow the progression of the disease. And despite significant developments in Alzheimer's diagnostics, the precise diagnosis remains problematic. In the Alzheimer's disease brain, besides the accumulation of amyloid and amyloid plaques and tau tangles, there is a significant neuronal loss. You can see on this image the significant neurodegeneration affecting the Alzheimer's disease brain. But the basal forebrain cholinergic system and the hippocampus, what you can see here circled with red, are early and very severely affected in Alzheimer's, but later in the disease, as you can see on the image, all brain regions are seriously affected by neuronal loss. Neurons form networks to communicate, but amyloid beta and tau and the significant neuronal loss as well and other pathological mechanisms in Alzheimer's disrupt this communication. The neuronal communication deficits and the additional neuronal death that follow further accelerate this communication problem. The places where neurons connect and communicate are called synapses. Each neuron can have between a few to hundreds of thousands of synaptic connections, which can be formed by itself and neighboring neurons and neurons in other brain regions. And synapses, as you can see here, on the image up here on the right corner, they are made up by presynaptic terminals and postsynaptic terminals. The presynaptic terminal is where the electric signal that comes through that neuron, the action potential is converted into chemical signal. And this chemical signal is a neurotransmitter that is being released. And GABA is the main inhibitory neurotransmitter in the nervous system, and glutamate is the main excitatory neurotransmitter responsible for stimulation. And they are critical for regulating the brain excitatory inhibitory balance. In Alzheimer's and most other neurodegenerative diseases, there is a significant remodeling of the neuronal networks affecting these neurotransmitter systems. And because maintaining the balance between excitation and inhibition is critical for normal neuronal function, and the components of these neurotransmitter systems are promising therapeutic targets. Besides the newly approved controversial amyloid beta targeting therapies, all currently available drugs for Alzheimer's are targeting the excitatory pathways. Memantine is one of the drugs, for example, and it has a small beneficial effect in people with moderate to severe Alzheimer's. The benefit is affecting thinking, the ability of carrying out normal daily activities, and the severity of behavioral and mood problems. So research is ongoing to develop more effective drugs targeting the excitatory, but also the inhibitory systems of the brain. During the past few years, my lab characterized the glutamatergic GABAergic system components in the human healthy brain and in the Alzheimer's disease brain, and also in animal models of this disease. We aim to identify better therapeutic targets by understanding the pathological changes. And in Alzheimer's, glutamate excitotoxicity is very well known and is directly linked with neuronal death. And the GABAergic system is critical. It plays a critical role, putting a brake of this overstimulation, the excitation, the increased excitation in the brain. So we need to learn more about the excitatory inhibitory remodeling in Alzheimer's, but there are already a few promising therapeutic targets that have emerged. And one of them is the alpha-5 subunit containing GABA receptors that have been identified as promising therapeutic targets. GABA receptors, they bind the inhibitory neurotransmitter GABA, and they are formed of 5 components called subunits. You can see them here. And one of these subunits is the alpha-5 subunits. And you can see on this image here, this is a section of a mouse brain. And you can see that the alpha-5 subunit containing the GABA receptors are labeled in the areas which are like very bright. And the brightest area is the hippocampus. And this is the brain's memory center, and makes this receptor perfect therapeutic targets because it has high levels in the hippocampus. And this is a mouse brain, but we also check the presence of the receptor in the human brain. So we examine the most common receptor subunits in the healthy and the Alzheimer's hippocampus, and we confirmed that the alpha-5 subunits containing GABA receptors are present in the human hippocampus. And interestingly, we found that it was highly elevated in the Alzheimer's disease cases. And previous research showed, importantly, that increased levels of these receptors in mice are sufficient to induce cognitive deficits. Therefore, we moved on and we expect that the pathological upregulation of the receptor in the human hippocampus will most likely disturb cognitive function in the Alzheimer's disease brain as well. So next, we established hippocampal neuronal cultures, which are brain cells growing in a dish, and we treated them with neurotoxic beta amyloid. Unexpected beta amyloid increased cell death and also increased alpha-5 subunit levels. But the alpha-5 subunit containing receptor targeting drug, alpha-5IA, reduced the cell death and also restored the receptor levels. These experiments suggested that blocking the alpha-5 GABA receptor activity could be a promising therapeutic approach. So Beatrice, a PhD student in the lab, also examined the tissue from mice that received amyloid beta injection into the brain, into the hippocampus, and then we treated the mice with a drug. We performed a battery of behavioral tasks to examine different aspects of learning and memory. And mice are curious creatures, and when you present them with new objects, they will explore them. So animals were habituated the day before the experiment by placing them in a testing box, and we presented them with 2 identical objects. The next day, then one of the objects was swapped, and the mice were presented with a familiar object and this new object. And they were introduced simultaneously. The healthy control animals, they recognized the new object and they spent more time exploring it. The amyloid beta administered mice could not discriminate between the 2 objects. And the mice spent approximately similar time with both objects. However, when we treated the animals with the drug, this prevented this recognition memory deficit induced by amyloid, and the animals were able to recognize the new object. We also examined the impact of the drug treatment and the activity of hippocampal neuronal networks. We used microelectrode array tools with hundreds of electrodes embedded in a recording chamber, where we placed the brain slice in this chamber, and then here you can see the recording electrodes, and the hippocampus just sits on the recording electrodes. Oops. Yeah. And so if we found that amyloid beta, when we treated the slides with amyloid beta, increased neuronal excitability, which was restored by the addition of the alpha-5 IA drug. She also showed that amyloid-induced reduced synaptic plasticity, and the drug was able to restore this. While these data are promising, and after some favorable clinical studies, unfortunately, long-term alpha-5A use was found to cause kidney toxicity. But despite this, the data presented here highlight that targeting the alpha-5 containing GABA receptors may be promising, an important direction for developing normal therapies for targeting Alzheimer's. And there are some promising drugs under development in this area, but drug development is a long and challenging process. Analysis across all therapeutic areas indicates that the development of new medicine, from target identification through marketing approval, takes around 15 years or longer. And this is particularly true for Alzheimer's targeting drugs. Another therapeutic approach to regulate the excitatory inhibitory balance is targeting transporters, ion transporters in the brain. And we know that maintaining chloride level in neurons is critical in regulating excitatory inhibitory balance. Targeting the KCC2 and KCC1 transporters that maintain chloride levels has been explored in several neurodegenerative, neurodevelopmental, and psychiatric disorders. Now, low chloride levels are critical to ensure that GABA will have an inhibitory effect. GABA can have excitatory effects instead of inhibition during development, and also disease conditions such as Alzheimer's due to this altered transporter activity. In this study, we injected amyloid beta into the mouse hippocampus again, and Patty demonstrated that amyloid beta can induce reduction around the ejection of amyloid and an increase in NKCC1 levels in the mouse hippocampus. What is the consequence of this? So what the consequence of altered transporter expression is increased intracellular chloride levels in the neurons and an altered excitatory inhibitory imbalance in the brain. How can we restore this? Bumetamide is a diuretic, and it has been repurposed to treat neurological conditions, and bumetamide inhibits NKCC1 and can reduce the increased chloride levels back to normal. And this is a promising approach, but research is needed to elucidate further effect of amyloid on KCC2 and NKCC1, especially in the human Alzheimer's disease brain, and to determine whether targeting these transporters will be therapeutically beneficial in Alzheimer's patients. So after the two drug approaches, I'm moving to some alternative ones, because the excitatory inhibitory balance can be regulated using non-drug approaches as well. Optogenetics is a technology that uses light-sensitive proteins delivered to specific types of neurons by viral vectors that can be activated by light, and this will lead to activation or inhibition of neurons. We have used these tiny fully implantable devices and in mice, and this allowed us to turn on or off specific neurons in the hippocampus. Here you can see a mouse performing a behavioral task, the novel object recognition task, what we mentioned, we used before for drug testing. And you can see the flickering of the optogenetic device. And importantly, we identified a stimulation protocol that can restore the cognition of the mice that have been injected with amyloid, and we have seen long-term memory deficits in these mice. And Currently, we are optimizing this stimulation protocol to achieve like long-term beneficial effects of the stimulation. But also, we are using this technology to understand better the excitatory inhibitory remodeling in the hippocampus in Alzheimer's. There are also non-invasive brain stimulation approaches that are currently being investigated to restore the excitatory inhibitory balance. And this is a developing field with great potential. Modulating the excitatory inhibitory balance is also possible with nutraceuticals. The food that we eat can regulate the excitatory inhibitory balance in the brain. And we know that one modifiable risk factor for Alzheimer's is healthy diet. Our research focuses on understanding the role of caffeine in Alzheimer's disease pathology and its potential in preventing the disease and restoring cognitive function. I'm running out of time soon, so I will not present like much data today, but I would like the audience to contribute to our research. Our caffeine survey is shortly closing. We are evaluating the caffeine intake habits in Ireland for the first time, and importantly, we are looking the connection with the cognitive function because we are included a cognitive failure questionnaire in this survey. And our preliminary data surprisingly show that Ireland is not a only tea drinking nation anymore because coffee and chocolate are equally important sources of caffeine intake. Caffeine emerged as a cognitive enhancer, but there is not enough evidence at the moment to suggest intake, and especially for dementia and Alzheimer's prevention. And we also need to explore better the risk related to the consumption and evaluate this better before we can suggest caffeine consumption, like changes in caffeine consumption habits. So in summary, there are many promising therapeutic targets, but the drug development process is challenging. And our goal is to understand better the pathological processes in Alzheimer's and other neurodegenerative conditions to further develop these treatment strategies. Over the years, many collaborators and students have been involved in this work, and we are very grateful for the support of people with dementia, and especially the brain donors, their families, the patient organizations, and funding bodies supporting this research. And thank you very much for your attention, and I will be happy to take questions in the end. Thank you, everyone.
Thanks so much, Andrea. That's great. It's really interesting presentation, very informative, and it's so interesting to see all the research that's happening from, we were a very talented bunch of PhD students, so thanks so much for that. I'm delighted now to introduce our next speaker, Professor Keith Murphy. Thanks so much for being here today, Keith. Keith Murphy is a professor of neuropharmacology at the School of Biomolecular and Biomedical Sciences in University College Dublin, where he runs the Neurotherapeutics Research Group. This group is dedicated to understanding brain function and dysfunction and the identification of novel drug targets for the treatment of neurological and neurodegenerative diseases, such as drug addiction, depression, anxiety, schizophrenia, cognitive decline, and multiple sclerosis. Professor Murphy's research uses several different approaches, including animal models of behavior, in vivo site-directed interruption of function, in vitro cell and organoid culture techniques, transcriptomics and proteomics, and computational neuroscience to develop these much needed new neurotherapeutics. And today, Keith is going to talk about investigating how memory works in search of better cognition enhancing drugs. The talk will introduce what we know and what we're finding out about how our brains store memories, and we'll explore how we might be able to use that knowledge to develop new drugs that can enhance and protect our memory functions against dementia and other brain diseases. So the floor is yours, Keith. Thanks.
Vanessa, thanks so much for the invitation. It's a pleasure to come chat to you all today. And thanks also to Andrea, who's done a lot of my heavy lifting for me in my introduction, so I can now quickly skip through some of the preliminaries. So I guess in the context of our work, uh, like Andrea's group, we're very interested, of course, in the brain and how it functions, and it is very much a circuit-based structure where the important and very complex functions that our brain performs really depends on the connections and circuits and how the synapses are working. And because it's a very complicated machine, as we all know, it can go wrong in very many different ways to cause problems with functions. And the types of disorders that we're talking about, memory disorders, revolve very much around one particular part of the brain called the hippocampus. And Andrea talked about the hippocampus quite a lot. And the hippocampus, I suppose, has for a very long time been known to be the part of the brain that's important for memory function. Uh, it's a subcortex, so it's underneath the surface, just here, this blue part. And one of the earliest indicators that it was very important for memory was this patient, HM, is a very famous patient in our area of research, because HM had epilepsy, and they performed a surgery to help HM's epilepsy because it didn't respond to any drugs at the time. But the part of the brain that they essentially did the operation on was actually the hippocampus. And when HM woke up, so you can see he had the surgery when he was 27 years old, and he lived for a very long time afterwards. He lived until he was 82. But from the point of surgery onwards, uh, HM could no longer actually form a new long-term memory. And so it was one of the first indications that in the human brain, the hippocampus is critically important for making memories. We know that's not where the information is stored. The information goes out here into the cortex, actually, and it's stored long-term in the cortex, but the hippocampus is critical for making the memories to begin with. And of course, this part of the brain is very badly impacted in diseases like Alzheimer's disease and dementia. And that's really why memory function is such a key symptom in those types of diseases. And so I suppose the last thing I'd note about memory, though, is that it's not just impaired in Alzheimer's disease and dementia. Actually, memory function is impaired in a whole range of brain diseases, which is very interesting. So it implies that memory function is quite fundamental, and it can be disrupted in the context of many different disorders, not just degenerative disorders like Alzheimer's, but also mental illnesses like schizophrenia and anxiety and depression and drug addiction. And my research group, I suppose, is very interested in coming at these disorders in terms of trying to find new treatments by exploring the memory function itself and seeing if we can find new and better targeting ideas or new targets or develop new drugs from knowing more about how the brain just actually performs normally in terms of memory function. And so I suppose then if we think about what do we know about memory function, happily, we now know a lot more than we used to. And by research in our group and many other groups around the world over decades now, we have now, I suppose, found out that memories are formed or stored in the brain because the connections get changed. So the synapses that Andrea described so beautifully in her talk, these connection points essentially are information contact points. And when new information gets stored in brain, especially in the hippocampus, we know that the number of connections temporarily gets bigger, so the nerves grow more new connections. And then there's a selection process, which brings us back to roughly the same number we started with, but they are different connections. So basically, this is the idea of the brain circuit reorganizing itself in order to store new memories. And the belief is that if we can understand how this process works and we can harness it and we can make drugs that can promote or improve this function, then we would be able to actually improve memory functions or rescue memory functions in disorders like Alzheimer's and other degenerative disorders, or indeed in the psychiatric illnesses like schizophrenia and depression. And so the question is, can we understand enough about this process and find new targets in order to create new drugs to help support memory function. So my group's been working on that for a very long time, and we use animal models to look at this question because animals are very robust models for learning. And you saw a novel object in Andrea's talk. That's a memory task as well. The memory tasks that we've been using are memory tasks like water maze. So that's just a spatial map. It's like basically a jacuzzi, and the animals have to find a submerged platform, and so it's a spatial mapping test. They have to form a spatial memory. And a different task is called passive avoidance. And in this task, the animals have a chamber where there's a light chamber and a dark chamber. They can move between them. But if they enter into the dark chamber, they get a little foot shock on the floor. And so when they're tested whether they remember this or not, they will tell you if they remember by staying out here in the light chamber and not going into the dark chamber. And so the interesting thing about these two things, this is an association memory. This is a spatial memory, but they're both dependent on hippocampus. And so we understand or know for a long time that when these types of memories are being formed, they're being formed in the by the hippocampus. And so that was the premise of our experiment. And we thought what we would do is we would try to find out what's happening in the hippocampus over the first 12 hours following learning either of these two tests. And ultimately, what we wanted to do was to understand at the molecular level. So in this experiment, we used gene expression. So the genes that are controlling the cell function and how much they're expressed. And we wanted to understand which genes were the most important genes for a memory event in hippocampus. So this is called transcriptomics, but basically what it's doing is it's measuring gene expression. And there are some incredibly sophisticated technologies available to us now to be able to do this. In fact, we can go all the way down to looking at transcripts in individual cells now. So it's become very powerful. But I was going to summarize a little bit of our research in this space, and tell you that when we did this experiment and we looked at the water maze, the spatial test, we discovered that over this 12-hour period, there are a very large number. So over 5,000 genes are changed at some point of the 12 hours. And in passive avoidance, the number is slightly smaller. It's 2,600 genes are altered at some point in the 12-hour period. And so that's interesting by itself. But the really interesting group of genes were the group of genes that were shared between the two lists. So there are about 1,500 genes that seem to regulate in the hippocampus, and they're totally independent of the type of memory being formed. And so we thought that if we were going to try to make new cognition enhancing or memory enhancing drugs, that these genes would be the interesting genes to study. So we went on to have a look at those genes. So 1,500 genes is a lot of genes to focus on in research. So we tried to find ways of narrowing down which ones we would look at. And one way we did that was by looking at clusters of genes. So a cluster of genes is a group of genes that regulate in exactly the same pattern over the time frame of the 12 hours. And so we found one particular cluster of genes, and these are just some of the members of the gene of this cluster, which all upregulate very quickly after the memory event or the learning event. And so they grouped together and they had the same pattern in the water maze and the passive avoidance. So they were being upregulated in the hippocampus, and they were being upregulated in very early part of the memory encoding process. And our question was, could we harness any of these as new drugs? So I'm just going to show you one of the drugs that we worked on, or one of the proteins we worked on called midkine, as an example of how you can take this fundamental knowledge and translate it into a potential new drug strategy. So with midkine, what we were able to show is that it is expressed very highly in the hippocampus. And so this red portion here is very high expression, blue is very low expression. And if you look at where is midkine in the brain, you'll discover that it's very heavily concentrated in the hippocampus. And so that immediately suggests that it has an important role to play in whatever the hippocampus does normally. And then this part of the experiment simply showed that the protein levels of midkine increase as well. So the first experiment was about gene expression, but now we can see that the protein expression levels go up as well. And that's important because protein has function in the cells, not necessarily the genes. So we knew that the amounts of midkine were increasing in the hippocampus following the learning event in the brain. But the final and important question is, of course, does that help us to build a cognition or a memory enhancing strategy? So we went back to the learning paradigms. So this is the water maze again. So in this test, remember, the animals have to find a submerged platform, so they have to make a spatial map. And if they're learning that well, they will get faster at getting to the platform each time you test them. So this is the first set of data where we were able to take two groups of animals, one control group, which are the white circles, and one group of animals were treated with midkine. So those are the blue circles. And what you can see is that the animals that get treated with the midkine, they're much faster at getting to the platform over the sessions. And then if you test whether the animals remember, so you go back a couple of days later and you can ask, take away the platform, just let the animals swim in the pool. And if the animals remember well, they should stay in the target quadrant. So that's this quadrant where the platform used to be. And you can compare it to the time in the other quadrants. And so animals that remember well should swim here where the platform used to be to tell you they remember where they're looking. And what you can see is that the midkine treated animals have a much better memory of where the platform used to be. And then to test whether this was a general memory enhancing property of midkine, we then looked at a different learning task called the odor reward association task. So in this task, the animals go in a box where there are 3 containers, and there's a sponge in each container with a different scent or a smell. And so we used almond, lemon, and coffee, so they're quite distinct smells, so the animals can tell them apart very quickly. And the test relates to the fact that only one of the containers has a food reward, and the animals learn to go to the correct odor in order to get their food reward. So it's an association task where the animals associate food with the odor. And the animals will learn that very quickly, and they'll get very quick at getting to the right odor. But the important point, I suppose, to note with the experiment is that if you treat half the animals with midkine and you test their memory, then the animals that were treated with midkine remember much better than the regular animals, the animals that didn't get the treatment. So in this positive association memory test, so very different to the water maze test, even in this test, midkine is a memory enhancing agent. And so this opens up the possibility that if we could harness this as a drug, then we might be able to develop a molecule which would be able to assist or enhance memory function generally. So I wanted to just broaden out the question about memory function, though, and treatments or memory enhancing drugs, and whether they would be useful to mental health disorders. So an interesting thing about mental health disorders like schizophrenia and depression and anxiety is that they have symptoms that actually are quite similar in some cases. And one of the symptoms that's very similar across those diseases is memory deficits or cognitive deficits. And so this means that memory enhancing drugs might be useful for treating these types of diseases, as well as they are useful, obviously, for treating something like Alzheimer's disease. And so with these diseases, the important thing to remember is that there's predisposing genetics and then environmental stressors that happen that alter brain development and change the circuits that are controlling behavior to basically cause these diseases or the symptoms to emerge for these diseases. And This is just to show you an interesting fact about these types of disorders, which is that they all tend to emerge in the teenage period of life into the early 20s. So it's a very consistent feature of these types of diseases that they emerge during that time window in life. And that's not an accident because this time window is associated with particularly parts of the brain that are maturing, like the prefrontal cortex, which is very important for behavioral control. And so we can study this idea using animal models of early life stress. So things that induce early life stress can disrupt brain development and cause symptoms that look like these mental disorders. And so you can understand the slides that are coming. I just wanted to say that so relating animal age to human age is kind of difficult. But roughly speaking, I'm going to show you some animal research which uses animals at 30, 40, and 60 and 80 days old. And so that you can understand how they relate to humans, the 30-day-old animals are early childhood in humans, and then 40 is early adolescence, 60 is late adolescence, and 80 is young adulthood in human terms. And so this later time point would be when you see symptoms of mental disorders actually appearing in human time. So if you keep those times in your mind, I'm going to tell you about a model that is used for mental health disorders called isolation rearing. And so rats and mice are very social creatures. They like living together and playing together. If you separate them out and have them only one in each cage, that's called isolation rearing. And this is a model that causes symptoms that you can see that relate to mental disorders. And so what you can see, this is a measurement of sensory processing, which is used a lot in this area. And it is used to basically show that the animal is not processing information from the world around them the way they should. So it's a very simple idea. If any of us hear a loud noise, then we tend to have a startle reflex or a jump. We jump. But if there's a small noise before the loud noise, then it's like a warning signal, and so the animals don't jump as much, and we don't jump as much. So this is a very useful paradigm or very useful measurement because it's exactly the same in humans as it is in the animals, and so it relates very well. If there's a deficit, it means that the brain is not processing information properly, and it will mean that the pre-pulse doesn't warn the system the way it should, and so the startle reflex remains very high. So basically, we know that in mental disorders, that appears in the mid-adolescent period into young adulthood. And you can see that the isolation-reared animals show that deficit. They don't have a good sensory processing system. So you can see that they exhibit a feature that you see in mental illness. So we've also been able to show that lots of other features of mental disorders are captured in the isolation rearing model. So, for example, the levels of glutamate, which Andrea was speaking to you about, so this is the excitatory neurochemical in the brain. The levels of glutamate are very low in the prefrontal cortex in the isolated animals. And that's interesting because you see that in humans with mental disorders. Also, the number of synapses, just the number of connections, is lower by about 10% in the cortex in the isolated animals. So they have lost connections. This is something you also see in disorders like schizophrenia, for example. And here we have the water maze again. So this is how fast the animals are getting to the platform. And here is the passive avoidance test. And these are both indicators. As you can see, the isolated animals don't perform as well on both these tests, and they forget the association in the passive avoidance test. So what that shows you is that the isolated animals have cognition or memory problems that are associated, again, with mental disorders. So all of this information shows that the model is a very accurate model for human mental health disorders. And so what we then did was if it's such a good model, then we can use it to ask about what's changing in the brain and whether we can find targets again that will help us to create new treatments that could help with these types of disorders, and particularly memory disorders within the context of mental health. So we did the same sort of experiment. We looked at different parts of the brain and we asked what genes are different between the normal animals, the social controls, and the isolated animals. So what we discovered was that there's a very significant number of genes altered between the social controls and the isolated animals. And we have genes like this first gene group here, which are immediate early genes. They're all transcription factors, so they control other genes. They are all profoundly low, too low, much too low in the isolated animals compared with the normal animals. And this is true even before those animals have symptoms. So that's a very interesting finding, because if you could fix this problem very early, in theory, you might be able to prevent the symptoms ever even emerging in these animals. But the cluster I wanted just to quickly end on and show you at the end brings us back full circle, because the genes which we found to be involved in normal memory, the midkind cluster, you'll remember, these are all suppressed or have much lower expression in the isolated animals compared with the normal controls. And so one could put two pieces of information together. In one hand, the isolated animals have memory problems. And on the other hand, we've now been able to show that these very important memory-associated genes are too low in the isolated animals. And could we actually be able to treat the symptoms of mental illness using something like midkind? And so this final graph over here shows you that, so the open circles are the normal animals, the sensory processing, the PPI. The square, these are the isolated animals, and so their sensory processing is lower than it should be. But if you take isolated animals, so these are the blue squares, and you give them midkind, so you treat them with midkind, then their sensory processing is perfectly normal. So that's one of the key symptoms in mental health, and it is essentially restored to normal by treating with something like midkind. And then the final two graphs are simply to make an interesting observation, which the best drug we use for psychosis, for example, which is clozapine, increases the levels of midkind in the brain as part of its function. So it may be that even the current drugs we use for treating psychosis may actually be working, at least in part, by normalizing these very important memory-associated molecules in the brain, and that then normalizes a lot of the behaviors that you see. So I'll wrap up at that point by observing then that we've been able to, and lots of other people around the world as well, have been able to now piece together a very detailed picture of what's happening in the brain during normal memory encoding. And we now have a whole list of different potential new targeting ideas that could be used to develop new drugs that can enhance memory or improve memory function. And that's going to be useful not just in dementia and Alzheimer's disease, but actually also in other mental illnesses, as I was showing you. And then I suppose I'd finish on the point that I now tell all of my undergraduate students, which is that this is now a very exciting time in developing drugs for the treatment of brain disorders after decades of very little progress. There is finally now a large number of new ideas and new drugs in the pipelines around the world, which, although, as Andrea points out, the timeline will be long because that's the nature of drug development. But there is now a wealth of new opportunities and new strategies that are being explored that we can, I think, have a really serious hope are going to improve the way we can treat dementias, degenerative disorders, and neuropsychiatric diseases over the next 10 to 15-year period. So I have to thank all of the folks who worked in the lab over the years on all of this work, and in particular the people who provide all the funding. And thanks all for your attention, and happy to take any questions that you might have.
Thanks so much, Keith. Really, really interesting again. Fascinating stuff. We have time for a few questions. So if you want to put any questions that you have in the chat box or in the Q&A box, please go ahead. There's one question here. I'll I'll read it out and then you can see who wants to answer, or both of you might be able to answer. I'm not too sure, but the question is around neuroinflammation. So neuroinflammation has been implicated in memory loss in Alzheimer's disease. What is your opinion on oligodendrocytes? Sorry, I'm saying that wrong.
You're very close though. Oligodendrocytes.
Close enough.
Oligodendrocytes.
Okay.
Yeah. No,
So
it's a very important field. Yeah.
I guess absolutely. Neuroinflammation is actually important for all degenerative disorders, and interestingly, also most mental disorders as well have inflammation components linked to them. So it's broadly important and interesting. And I suppose so in the brain, it's astrocytes and microglia would be traditionally the cell types that mediate inflammation in brain tissue. And they have a huge role to play in Alzheimer's, and many of Andrea would know much better. Oligodendrocytes, though, are the cells which do the insulation around the axons. So they're a slightly different group. They are damaged in diseases like multiple sclerosis by inflammation. But oligodendrocytes may be slightly less important players in things like Alzheimer's disease, I suppose, would be the way
But there
to
is
look at
like
that.
lots of attention now on them, like luckily, because we are just learning, that they also like release like important like immunomodulatory molecules, and then just like for glia and astrocytes during the past 5-10 years, like big transcriptomic studies have been performed and classified them into different activation states. So that's happening for the oligodendrocytes as well. And recently it has been shown that they might be contributors even to amyloid production. So we need much more research on these glial cells, because they are directly involved in the regulation of excitatory inhibitory balance as well, by like clearing the neurotransmitters from the synapses. But then as Kate said, like even just the transmission of the signal through the axon, like those myelin sheets are like critical, and oligodendrocytes are needed for that. And what is very interesting, Kate, you mentioned, like some of the drugs, like we don't know even some of the effects. Donazepam that is prescribed for Alzheimer's, actually it has an effect on oligodendrocyte generation and the remyelination process. So yeah, important field. Yeah.
It's very interesting. I'm not from a neuropharmacology background myself. You think, God, it's so interesting to hear all this, because it's absolutely fascinating. I just wanted to out of my own curiosity, you were both speaking about obviously the development of new drugs and, the long process it takes. And, but Keith, you finished on a very positive note, I suppose, very optimistic note that it is a very exciting time. So this might be difficult to answer, but where would you see this field in like 5 to 10 years' time? Will it be transformed, or will we still be kind of moving forward in a positive way? What can we expect, I suppose?
Yeah, look, that's a great question. It's I always like to be realistic rather than, exaggerating where we're going to get to. And I think Andrea's point is very important. It is a long process. It's long in any disease state, but the neuro disorders tend to be even that little bit longer, I suppose. But I'm coming back to my finishing point. Um, if you were to let me answer the question this way: if you looked at where we were 15 years ago and compared it to where we are now, we know so much more about just disease processes and what's going wrong and all the different cell types that are involved. Uh, like the landscape has really transformed really dramatically, and I think everybody in our field is
Yeah.
of the same view and equally excited about those really huge steps forward in just understanding stuff. And that is ultimately, it has to then start to translate into new therapeutics. Certainly the pipelines are now much richer in terms of opportunities and different strategies. The challenge is going to be how many of those are going to fail when they get to the real test bed in clinical settings and clinical trial settings. That's a really hard one to predict. The attrition rate tends to have been very high. And but all of that said, I think we can realistically expect 10 to 15 years from now to have significantly better therapeutics clinically approved in most of the brain disorders. That would be my opinion.
Yeah.
Thanks so much, Andrea. Do you want to?
I agree
Yeah.
with you, Kate, and I just would like to emphasize that probably like the developments in diagnostics, that
Yeah,
will be a game changer because like 10 years ago, we were really running the trials with those who have been really in advanced stages
yeah,
of dementia, different type of dementias, including Alzheimer's. And now with better diagnostics, we will be able to target the specific population we want to, and also the clinical trials and all the studies are becoming more international. That's a big development as well. So it's not multi-center big trials. That's very important. Yeah.
yeah, yeah. And I suppose maybe just to wrap up that there is also going to be a shift, certainly in the mental disorder space. There's a clear shift to developing therapies that are proper disease modifying or preventative strategies. So we all sort of know that if you can stop the brain getting so badly damaged or so badly dysregulated, that would be a better strategy, right? So don't let it get as damaged. Don't let the circuits get so dysregulated. Uh, so if you can intervene early, and there are strategies around that type of idea, if you can intervene early, you can in theory stop the circuit getting so unhealthy. And in fact, symptoms will either be much reduced or possibly not even emerge at all. And so that's again, that's a step beyond. We're probably maybe 15, we're probably 20, 25 years away from strategy like that. But that would be a huge step forward. Uh, so it's, yeah, so I think there's plenty of space for optimism.
Well, we're 1 minute now to 12, so we better wrap it up. But that's a really nice note to wrap it up on, a positive note that, while it might still be some, bit of time left, we're still going in the right direction. And like you said, there's lots in the pipeline. So that's a great thing to end on. So before we finish up, I just want to let you know that this webinar has been recorded and it'll be available on the DRNI YouTube page. It should be up in the next couple of days, and you can also find all our previous webinars recorded there. So just to finish up, then I want to say on behalf of DRNI, thanks so much to everyone who attended. Huge thank you to our wonderful speakers, Andrea Kwakowsky and Keith Murphy. It's been really fascinating to hear about all the different research you're doing, and you just learn so much about neuropharmacology and the brain. So thanks so much for your time today and for your excellent presentation. And everybody enjoy the rest of your day. Thanks so much.